Welcome to this educational video on cladistics, a modern approach to biological classification. Cladistics is a revolutionary method that helps scientists understand how different organisms are related to each other through evolutionary history. Instead of grouping organisms based solely on how they look, cladistics focuses on their evolutionary connections and shared characteristics that have been inherited from common ancestors. Cladistics is a method of classification that groups organisms based on evolutionary relationships and shared derived characteristics. This means that instead of simply looking at physical similarities, scientists examine traits that organisms inherited from their most recent common ancestor. These shared derived characteristics, also known as synapomorphies, are key features that help us trace the evolutionary path of different species and understand how they are truly connected in the tree of life. To understand cladistics, we need to learn four essential terms. First, a clade is a group consisting of an ancestor and all of its descendants, representing a complete branch of the evolutionary tree. Second, a cladogram is a branching diagram that visually shows these evolutionary relationships. Third, a synapomorphy is a shared derived characteristic that is unique to a particular clade, helping to define that group. Finally, a node is the branching point on a cladogram, representing the location of a common ancestor where different lineages split apart. In cladistics, we distinguish between three types of characteristics. Ancestral or plesiomorphic traits are those inherited from distant ancestors and are shared broadly across many groups, so they do not help us identify specific evolutionary relationships. Derived or apomorphic traits are new characteristics that evolved in a specific lineage, making them more useful for classification. Most importantly, shared derived or synapomorphic traits are derived characteristics that are shared by members of a particular clade. And these are the key features we use to define evolutionary groups and build accurate cladograms. A cladogram is a powerful tool for visualizing evolutionary relationships. It consists of branches that represent different lineages of organisms. Each branching point, called a node, represents a common ancestor shared by the groups that branch from that point. The organisms at the tips of the branches are the species we are comparing. Along the branches, we mark the shared derived traits, or synapomorphies, that evolved at specific points in evolutionary history. By reading a cladogram, we can trace back through time to see how different species are related and which characteristics they share due to common ancestry. In cladistics, not all groupings of organisms are considered valid. A monophyletic group includes an ancestor and all of its descendants, forming a complete clade. This is the only type of grouping that cladistics recognizes as valid because it accurately represents evolutionary history. A paraphyletic group includes an ancestor and some, but not all, of its descendants, which makes it incomplete and therefore invalid. A polyphyletic group contains members that come from different ancestors and do not share a most recent common ancestor, making it completely artificial. Only monophyletic groups truly reflect the natural evolutionary relationships among organisms. Constructing a cladogram follows a systematic four-step process. Step 1. Identify the organisms you want to classify and compare. Step 2. Select the characteristics you will examine and determine which ones are derived traits versus ancestral traits. This step requires careful analysis of when each trait first appeared in evolutionary history. Step 3. Group the organisms based on their shared derived characteristics, placing those with the most recent common derived traits closer together. Step 4. Construct the branching diagram with nodes representing common ancestors and label the branches with the derived characteristics that define each split. This process creates a hypothesis about evolutionary relationships that can be tested with additional data. Let us examine a classic example of a vertebrate cladogram. Starting from the base, fish represent the earliest vertebrates. At the first node, we see the evolution of jaws, a key innovation. Moving up, amphibians branch off after the evolution of lungs, which allowed vertebrates to breathe air. Next, reptiles split from the main line after the development of the amniotic egg, which freed vertebrates from dependence on water for reproduction. Finally, mammals are distinguished by unique traits such as hair and mammary glands. Each of these derived characteristics marks an important evolutionary transition, and by following the branches, we can see how these major vertebrate groups are related through common ancestry. Cladistics offers several important advantages over traditional classification methods. First, it is based on evolutionary relationships rather than superficial similarities in appearance, which means it reflects true biological history. Second, cladistics provides testable hypotheses about ancestry that can be confirmed or revised as new evidence becomes available, making it a scientific approach. Third, the methodology is objective and repeatable, allowing different scientists to arrive at the same conclusions when analyzing the same data. Finally, and most importantly, cladistics reflects the true natural history of life on Earth, organizing organisms according to their actual evolutionary connections rather than arbitrary groupings based on convenience or appearance. Let us review the most important concepts from this presentation. First, cladistics groups organisms by shared derived characteristics, which are traits inherited from a common ancestor. Second, only monophyletic groups, which include an ancestor and all of its descendants, are considered valid clades in this system. Third, cladograms show evolutionary relationships based on when traits evolved, not on overall similarity in appearance. Finally, this method reflects the true natural history of life, providing us with the most accurate picture of how organisms are related through billions of years of evolution. By understanding cladistics, we gain insight into the magnificent story of life on Earth.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.